Okay. Before I start, I just want to mention that I have a stutter, so sometimes my words don't come out as fluent, but I'm not dying or anything. I'm fine, just so you know. Hi everyone. My name is Emma. I'm a second-year PhD student at the Mind-Body Research Group at the University of Leuven, and I'm going to explain to you a part of my PhD where I will investigate the role of inflammation and stress physiology in major depressive disorder. More specifically, I will investigate into biomarkers for treatment response. The way that we are treating and diagnosing depression now is based on a diagnostic and statistical manual 5, but this method is often, this symptom-based method is often ineffective due to its lack of biological basis. And that's where the research domain criteria initiative comes forward, which offers a more dimensional framework by incorporating neuroscience, behavioral science, genetics, to identify biomarkers that could classify patients with depression into more homogeneous subtypes, which can then lead to better and more personalized treatments. In the past, there have already been immune markers and stress physiology markers described in patients with MDD, but till today, their interplay hasn't been investigated yet. So first, I wanted to show you what we already know about the neuroimmune axis when during acute stress. So when a stress presents, the immune system and the autonomic nervous system work closely together to maintain homeostasis. So when a stress presents, the autonomic nervous system will activate the sympathetic nervous system, and this leads to the release of stress hormones. These stress hormones will then bind on immune cells, and this will enhance the immune activation during that stress response. When the stress is then gone or alleviated, the parasympathetic nervous system is activated to counteract the effect of the sympathetic nervous system by releasing the neurotransmitter acetylcholine, and this will reduce the immune activation back to baseline levels. But when the stressor persists, so for example, with people that have chronic stress, the sympathetic nervous system stays activated without the normal counteracting effect of the parasympathetic nervous system. This will lead to an immune disbalance and to inflammation, which is mediated by the release of pro-inflammatory cytokines. These cytokines can then lead to a blood-brain barrier leakage and can enter the brain to cause neuroinflammation. And then neuroinflammation can lead to impaired cognitive function and decreased neural plasticity. And those things can lead to a whole range of mental disorders, including major depressive disorder. So what do we already know about the neuroimmune axis in clinical research? So research indicates that a subtype of people with MDD exhibit an immune dysfunction based on, let's see, C-reactive protein levels. C-reactive protein is a protein that is made by the liver when inflammation is present. We also know that both the immune system and MDD is associated with an autonomic dysfunction. So more specifically, they saw that higher inflammatory markers are associated with a lower heart rate variability, and that more severe depressive symptoms is also associated with a lower heart rate variability. Then, if we look at the immunomediated subtype of the depression, there are some trials that looked, okay, there is an inflammation in patients with the depression. Can we treat the depression by giving anti-inflammatory medication? And there are already quite some studies, and the two that had the best results are minocycline and celecoxib. So those are both anti-inflammatory medication that reduce inflammation and also reduce the depressive symptoms in the patients. But despite that the autonomic nervous system has been both shown to be associated with the immune system and with NED, there are till today no studies that look at the moderating effect of the autonomic nervous system in the treatment response when giving anti-inflammatory medication to these patients. So this will lead me to my objectives and my hypothesis. So I will investigate the association between HRV and inflammatory markers in individuals with MDD, and we hypothesize that individuals with lower HRV indices and higher inflammatory markers exhibit higher levels of depressive symptoms. I will also investigate whether individual differences in HRV and inflammatory markers are predictive to the treatment response. And we hypothesize that high inflammatory markers and lower HRV indices at baseline are associated with a higher response to anti-inflammatory medication in patients with MDD. And we will investigate longitudinal changes in HRV inflammatory markers and depressive symptoms during their treatment. And we hypothesize that longitudinal changes in these markers, in inflammatory markers, will be significantly correlated with changes in HRV and their symptoms. So how am I going to investigate my objectives? I will collect that data for the INSTA-MD study, which is an acronym for inflammation-based stratification for immune target augmentation in major depressive disorder. And in the study, we will investigate the effects of minocycline and celecoxib and compare them as an add-on therapy in patients with MDD. So what is the design? So in total, we will recruit 240 participants with major depressive disorder that are on stable antidepressant treatment for at least 4 weeks. Then we will stratify those patients based on their CRP level, so an inflammation-based stratification, and then in a low and in a high inflammation group, so people that have 3 milligram per liter CRP in their blood, they will be in a low inflammation group, and the patients with 3 or higher will be in a high inflammation group. Then both the high and the low inflammation group will be randomized in one of the 3 treatment groups, which are minocycline, celecoxib, or placebo. And so in total there are 6 treatment groups, and they will each treatment group will receive this one of these 3 medications for a total of 12 weeks. And then during multiple time points, we will measure quite some outcomes. So we will measure immune markers, for example, immune cells and cytokines. We will measure their autonomic functioning. We will also measure activity levels and sleep. Of course, we will measure their symptoms, but also other their depression symptoms, but also other subjective symptoms with clinical assessments. Okay, so that was my presentation, and if anyone has questions, feel free to ask.
Thank you, Emma. Are there any questions?
Thank you. Really interesting. I had a question about possible comorbidities in these patients, like cardiovascular pathologies that can co-occur with depression symptoms.
Yes, that's actually quite some research about it. They even call it like the immunometabolic type of depression, because there is a subtype of depression that's like, as I was saying, like high inflammation markers, but also they have some kind of hormones like ghrelin, leptin. They also have higher cholesterol. And these patients also don't respond well to the convenient antidepressants. So and those are things we are also measuring. So we measure triglycerides, we measure glucose, so we measure all the metabolites to see if maybe the immune markers will be linked with those metabolites. But it's good question. They're also really looking into that. Yeah.